Hello everyone. Now we are going to discuss about the life cycle of the Plasmodium in man. That is asexual life cycle of Plasmodium in man. Before going to the asexual life cycle of Plasmodium in man, first we have to know about Plasmodium. What is Plasmodium? As we know, Plasmodium is a parasite which is responsible for the causation of the disease known as malaria. Here Plasmodium, if you are taking actually Plasmodium vivax is a common parasite which we are taking responsible for causing the malaria. Plasmodium vivax is always digenic parasite which completes its life cycle in two hosts, that is sexual life cycle in the female Anopheles mosquito, that's why we are calling as a primary host, asexual life cycle in the human being, hence we are calling it as a secondary host here. And then apart from that thing, what is happening here? Intracellular parasite. The Plasmodium which we had, the Plasmodium always we are saying as what? We are saying it as a intracellular parasite. Intracellular parasite, it means what? Which lives within the cells. Red blood cells, liver cells, all these things, hence we are calling as an intracellular parasite here. If you see about the different types of the Plasmodiums here, Plasmodium vivax we had, it always causes the benign tertiary malaria, and the Plasmodium falciparum, which always causes the malignant tertiary malaria. Benign tertiary malaria is not harmful, malignant tertiary malaria is harmful, caused by the Plasmodium falciparum. And Plasmodium ovale will cause the mild tertiary malaria, and Plasmodium malaria will cause the quaternary tertiary malaria here. This is about the different types of the Plasmodium parasites we are saying. If we go for the introductions, that means which scientist has been contributed for the discovery of the parasites, parasite Plasmodium, all these things here. Charles Leverson is a scientist which he first said that actually, which he first identified the Plasmodium parasite in the blood of the malaria patient. Then Sir Patrick Manson. Sir Patrick Manson, while he is conducting the experiments on the elephantiasis disease, he said that actually the spread of the malaria is because of the mosquitoes. Then very important thing, the very important scientist who has been contributed a lot for the study of the malaria, particularly Sir Ronald Rose. Sir Ronald Rose, while he is working in the Secunderabad, what he has done actually, he discovered the oocyst stage of the complete life cycle of the Plasmodium in the female Anopheles mosquito stomach wall on August 20, 1902, right? Particularly August 20, 1902 particularly. And that's why, as he discovered on that day, August 20 is celebrated as a World Mosquito Day, right? And then April 25th is celebrated as a World Malaria Day, particularly. And here, what is happening here actually? And he conducted all the Ronald Rose conducted all his experiments on the malaria parasite, that means Plasmodium parasite in the Secunderabad only. In the honor of his completely research work here, the fever hospital in the Tarnaka is named as a Sir Ronald Rose Fever Hospital here, right? This is about the introduction of the particular, and then particularly Garnham is a scientist, he has been also contributed for the Plasmodium life cycle particularly here. And then this is about the introduction of the Plasmodium life cycle. If you are going for the asexual life cycle of the Plasmodium in man here particularly, asexual life cycle mainly consisting of what? Here 3 steps mainly: pre-erythrocytic cycle, exoerythrocytic cycle, erythrocytic cycle. 3 steps are main important in the asexual life cycle here. Here particularly in the pre-erythrocytic cycle, how this cycle is starting? The infective stage of the malaria always we are calling as a sporozoid stage. The sporozoid stage is the infective stage of the malaria, right? Of the Plasmodium here. 2000 sporozoids are released into the human body whenever the mosquito bites. Female Anopheles mosquito bites, whenever it is happening, what is happening here? 2000 sporozoids are entering into the blood of the particular, that means saliva of the particular human being cell, that means human body here. Out of the 2000 sporozoids, what is happening? The sporozoids are entering into the liver cell first, right? And by this thing we are calling it as a pre-erythrocytic cycle, hepatic schizogony. Schizogony, it means the method or the process of formation of the schizons, we are calling as schizogony here. Short and Garnham are the scientists mainly, they first identified the hepatic schizogony. In the hepatic schizogony, what is happening is the sporozoids which are there, first they are entering into the liver cells. In the liver cells, what is happening? First they are developing into tropozoite. Tropozoite is the feeding stage, right, particularly. And here what is happening? The tropozoite, by acquiring all the requirements here, it turns into larger schizont. The schizont undergoes the first division, and many schizons are forming here. That phenomena we are calling as a schizogony. And the first stage after the division we are calling as what? Cryptozoites are formed in the pre-erythrocytic cycle here, right? And remember the point here actually. Here particularly for the completion of this complete pre-erythrocytic cycle, we require 8 days here, right? For the formation of the cryptocytes here particularly. And pre-patent period we are saying. Pre-patent period, it means what actually? The period, the time gap between the first entry of the sporozoite and the second appearance of the sporozoite as a cryptocytes. That is the pre-patent period, right? Now the cryptocytes which are formed in the pre-erythrocytic cycle, they again enters into what? They again enters into the this thing again, the exoerythrocytic cycle. Here what is happening here particularly? The cryptocytes are entering into what? Entering into the other than the red blood cells here. When the cryptocytes are entering into other than the red blood cells, the same thing. First, wherever you go, the first stage is always tropocyte stage here. Tropocyte stage again acquires the required things and they converts into scission, larger cell, that is scission. Scission undergoes again, divides and forms schizogony. This after this schizogony again what is happening? Here they are converting into merocytes. Here the scission which is there in the exoerythrocytic cycle, it forms two types of the merocytes mainly actually. Here macro two types of the cryptocytes here. Macro metacryptocytes we had, then micro metacryptocytes we had. Macro metacryptocytes which we had again, they again attacks the liver cells, they continues the again the cycle continued here. Micro metacryptocytes which it is formed here actually, they attacks the RBC cells. Then whenever the micro metacryptocytes which are smaller, micro it means smaller, macro it means larger, when they are attacking the RBC cells, from here the erythrocytic cycle starts. We are also calling it as a Golgi cycle because Camellia Golgi has been first identified the erythrocytic cycle. And this exoerythrocytic cycle completes in 2 days here, right? Now we are starting with the cryptocytes. Micro metacryptocytes, when they attack the RBC cells, what is happening? First again there it is turning into what? Tropocyte. Same thing, whatever the stage you take, tropocyte. First stage we are calling as tropocyte here. The tropocyte which it is formed here actually after the attacking of the micro metacryptocyte to the RBC cells here, tropocyte changes into what? Signet ring stage here. Signet ring stage, it means what actually? The nucleus, particularly the tropocyte, will arrange as a signet ring, as a ring structure, hence we are calling it a signet ring stage. This signet ring stage, by acquiring the pseudopodia, it turns into what? Amoeboid stage. As we know, Plasmodium is non-motile because if you see the classification of the Plasmodium, particularly actually what is happening here? Plasmodium belongs to phylum Protozoa and then subphylum Sporozoa and class Telospora. Sporozoans are the organisms in which locomotor organs are absent here. Particularly here, temporarily it is developing pseudopodia. After some time, what is happening? Pseudopodia is lost again. Now, amoeboid stage gives rise to what again? Scission stage here. This scission stage, around the scission stage, what is happening here? Around the scission stage, particularly here, what is happening here? Schaffner granules are developed here. Here, Schaffner granules or as hemozoin granules which are there. What is happening here actually? Here the hemoglobin which is there, as it is completely taking place in the RBC cells actually, what is happening here? Plasmodium parasite, they attacks the hemoglobin. In the hemoglobin, they attacks on the heme part here. That heme part are converted into small red colored droplets we are calling as what? Hemozoin granules. By the formation of the hemozoin granules, what is happening here? Symptoms are appearing here. Right? Symptoms are appearing. What is the incubation period? Incubation period, it means first entry of the sporozoite and then appearance of the symptoms. That period we are calling as what? Incubation period. It is up to 10 to 14 days here. By the formation of the hemozoin granules and the Schaffner granules only, what is happening? The symptoms are appearing in the malaria stages. Symptoms, it means what? Cold fever, chills, all these symptoms they are appearing by the formation of the hemozoin granules, Schaffner granules and merocytes only here. Now, the hemozoin granules which are formed here, inside the hemozoin granules, what is happening? The scission divides and forms the merocytes. Now, merocytes which are there, they are released outside by the rupture of the wall particularly, right? Then, after the release of the merocytes, what is happening? The merocytes are developing into what? They are developing into the next stage, last stage of this particularly asexual cycle in the human beings, that is actually male gametocyte and female gametocytes are developed here. By this complete thing, what is happening here? At last, by the asexual life cycle of the Plasmodium in the human being, lastly what we are getting? Male gametocyte we are getting, female gametocyte we are getting. Starting is tropocyte, last stage is what we are getting here? Male and female gametocyte here. This is complete about the, it is complete thing about what? It is complete thing about the asexual life cycle in the man of by the Plasmodium, right?